So today we are going to discuss about the shoot apical meristem. Today we are going to discuss about the shoot apical meristem. So the meristem, which is present at the tip of the shoot, is called as shoot apical meristem, or in short form we can call it as a SAM. So if we'll talk about what is the construction, how this shoot apical meristem look like, or what are the size and the different shapes of this shoot apical meristem. First, we have to say that this shoot apical meristem has a set of undetermined cells, or we call these undetermined cell as a meristem cells. These meristem cells are having ability to grow or to differentiate into different types of tissues. That is, it is able to go for indeterminate growth. To its like SAM apical meristem, there are leaves which forms the close proximity with this apical meristem, or these apical meristems are also called as apical initials. So if we will see in the diagram, there at the center we are having apical meristem, then there are small leaf like structure. These leaf like structures are called as leaf primordia. These are actually the young leaves which covers or overlaps our shoot tip. These leaf primordias are the youngest leaves, or we can call these as they are the first leaves which are formed via the leaf primordia. So if we'll talk about the size sorry, if we'll talk about the size of this shoot apical meristem, it's in the range of 100 to 500 micrometer. It's in the range of 100 to 500 micrometer. And if we'll simply talk about the diameter of the apical meristem, it is measured up to 100 micrometer. And if we'll talk in lengthwise, then it is 250 micrometer. To this leaf apical meristem, it contains 1 to 3 leaf primordia in addition to the apical meristem. The size, shape, and the organization of the shoot apical meristem depends on various parameters. Like if we will take a plant, if we have to see the size of the shoot apical meristem or the shape and the organization of the cells of this shoot apical meristem, it depends on number of factors. Like first, the shape, size, organization will vary between the species of the same genus. Means simply बोल सकते species to species the size, shape and organization vary करता है. Also it depends also on the developmental stage कि हम लोग किस developmental stage पे SAM को observe कर रहे हैं. Or we can say the growth conditions, ये hot climatic plant है या cold climatic plant है, it depends on that also. So in short we can say the size, shape or organization varies, which depends on many parameters like species, which plant species we are studying, so it will vary, and at what developmental stage of the plant we are observing this shoot apical meristem and the growth conditions. As we have seen, largest shoot apical meristem in vascular plants. We have seen largest shoot apical meristem in vascular plant, and the best example is cascades. And the best example is cascades, and it has been measured up to 3 millimeter in diameter. It has been measured up to 3 millimeter in diameter. This was the one like largest. If we go on the extreme side, if we will see the shoot apical meristem in case of Arabidopsis thaliana, where there we measure this shoot apical meristem is less than 50 micrometer in diameter. In Arabidopsis, we are having less than 50 micrometer SAM in diameter, and it contains only few dozens of cells. Means साइज भी छोटा होता है अपेकल मेरिस्टम का, और उसके पीछे दाएं बाएं बहुत लिमिटेड नंबर ऑफ सेल्स पाई जाती है. Even we can say that in a given species we are having significant, मतलब एक particular species में भी वेरिएशन है सैम है. मीन्स डिपेंड्स ऑन द टाइम कि किस टाइम पे हम लोग सैम को शूट अपेकल मेरिस्टम को ऑब्जर्व कर रहे हैं. Even जो ये हमारा shoot apical meristem, it varies in size. Sometimes it is flat or it's mounded type, like you can see in the diagram. The variation, जो ये हमारी variation है किसमें, shoot apical meristem में, this is mostly associated with the successive rounds of leaf initiation. Means कितने सारे leaf primordia बनेंगे, उस पर भी हमारा शेप जो है किसका, सैम का, वो डिपेंड करता है. एंड द सेल्स विच आर फ्लैंकिंग टू दिस सैम, मीन जो सेल्स दाएं बाएं हैं हमारे शूट अपाइकल मेरिस्टम के, वो भी डिटरमाइन करती है. और जो हमारे दाएं बाएं की जो सेल्स है वो फिर बन जाती है, कमिट हो जाती है टू डिटर्मिनेट फेथ. जो ही शूट अपाइकल मेरिस्टम ग्रो करता है, जो उसकी नीचे वाली सेल्स होती है वो फिर फिक्स हो जाती है कि इस सेल्स को क्या क्या बनाना है. इवन हम लोग वी कैन सी द वेरिएशंस रिलेटेड टू द ग्रोथ रेट, और हम सिंपली बोल सकते हैं दिस वेरिएशन, दिस द साइज और द शेप डिपेंड्स ऑन द ग्रोथ रेट ऑफ द प्लांट, इंक्लूडिंग जब एक शूट अपेकल मेरिस्टम को फ्लावर बनाना है या तो उस शूट अपेकल मेरिस्टम को बर्ड शेप में रहना है. अगेन आई विल रिपीट दिस पॉइंट. ये जो शूट अपेकल मेरिस्टम है इसका साइज शेप वैरी हो जाता है, तो फिर किस पे ये डिपेंड करेगी, सीजनल डिपेंडेंट भी रहता है. ये जो साइज एंड शेप है ये सीजनल डिपेंडेंट भी रहता है. लाइक अगर इस शूट अपेकल को मेरिस्टम को फ्लावर बनाना है तो ऑब्वियसली this shoot apical meristem or the meristematic activity at this point will get lost if there is a formation of the flower, or sometimes these buds or the shoot apical meristem doesn't grow, it remain in a dormancy stage, so obviously there will be the different shape and size. When we see this shoot apical meristem under microscope, or we can say after observing shoot, after examining the shoot apical meristem under the microscope, it's observed that there are different zones. They reveal different zonation. In the center, we are having central zone, Caesar. This Caesar region is, we can say, it's similar to the quiescent center of the roots, which we have discussed in our previous lecture. This central zone contains the cluster of infrequently dividing cells. Means continuously इस central region से cells बाहर आ रही है. ठीक, means it acts as a quiescent center as in case of roots. Then to this central zone we are having flanking regions. Means दाएं बाएं से flanking regions है. These regions are called as peripheral zone. जो ये हमारा पेरिफेरल जोन है, they are having dense cytoplasmic cells. They are having cells which are dense in their cytoplasm, and are more frequently dividing cells, which later are able to form the leaves. The central position, the rib zone. This rib zone is in close association with the central zone, and from this rib zone we are able to form, our plant is able to give rise to different internal tissue of the stem. If we will, even these there are these regions, but in these regions there is a difference in frequency of division. मीन जो ये रीजन्स है ये रिदमिक एक ही रिदम में सेल डिवीजन नहीं करते हैं, वो डिफरेंस दिखाते हैं इन फ्रीक्वेंसी ऑफ सेल डिवीजन. If we will see the example in case of angiosperms, there are some cells which are superficial. These superficial cells reflect in they मतलब वो रिफ्लेक्ट करते हैं किनको, layered organization को. Means if we will see the superficial cells of angiosperms, from these cells we reflect there is the difference in the organization of the cells, and sometimes we call these मतलब dividing cells अलग अलग हम तो हम लोग ऑब्जर्व कर सकते हैं कि कुछ अलग अलग चीजें मतलब लेयर जो है वो अलग अलग कुछ कुछ चीजें सेल्स बना रही है. इफ वी विल सारी सेल्स को अगर मतलब कलेक्टिवली लिया जाएगा, these cells are collectively called as tunica. These cells are collectively called as tunica. The tunica is this tunica is defined by the consistent pattern. Like ये क्या है longitudinally ये फिर divide हो जाते हैं. So that type of division is called as anticlinal cell division. जो ये हमारी tunica cells है, they form the consistent pattern, means एक similar pattern बनाते हैं. और जो similar pattern है, this is observed by the anticlinal cell division, which are able to produce tissue of uniform thickness, and we are able to observe these cross sections under microscope very easily. अगर ये cell same thickness की बना रहे, सेम टाइप ऑफ सेल डिवीजन हो रहा है, मीन्स वो सिमिलर टाइप्स ऑफ सेल बनाएंगे. सो बाय रिकॉग्नाइजिंग दीज cells under the microscope we are able to recognize that they are tunica cells. And the cells which are interior to this tunica are called as corpus. मीन्स एक तो बाहर वाला रीजन है, सुपरफिशियल लेयर है, उसमें क्या है एंटीक्लोनल सेल डिवीजन हो रहा है, सेल्स की कंसिस्टेंसी है और यूनिफॉर्म थिकनेस है सेल्स की. जब हम इस सेल्स के अंदर देखेंगे, अंदर वाले साइड को, इंटीरियर टू दिस ट्यूनिका रीजन, दिस रीजन इज कॉल्ड एज कॉर्पस. दिस इज कॉल्ड एज कॉर्पस. इस कॉर्पस में खासियत है, देयर इज वेरिएबल सेल डिवीजन. देयर इज वेरिएबल सेल डिवीजन. जब वेरिएबल सेल डिवीजन है, ड्यू टू दिस वेरिएबल सेल डिवीजन it leads to increase in the tissue volume. It leads to increase in the tissue volume. जब variable cell division हो रहा है उसकी वजह से क्या हो रहा है, जो टिश्यू का वॉल्यूम है वो इंक्रीज हो रहा है. जो ये शूट अपाइकल मेरिस्टम है, ये शूट अपाइकल मेरिस्टम हमें डिराइव होता है विद द स्मॉल नंबर ऑफ अपाइकल इनिशियल्स. एक छोटे छोटे सेल्स होती है जिनको हम लोग बोलते हैं इनिशियल्स. So the initials which are present at the apex of the shoot are called as apical initials. So in other words we can say that shoot tissue are derived from the small number of apical initials. After treating these apical initials with the colchicine, we all know what is colchicine chemical. So what it induces? It induces the polyploidy cells. What is polyploidy? It increases our nuclear volume. Means DNA double हो रहा है, but they are not, this DNA is not divided into the daughter cells. तो उसकी वजह से क्या होता है, हमारा न्यूक्लियर वॉल्यूम इंक्रीज होता है, जिसकी वजह से हमारा सेल साइज भी इंक्रीज हो जाता है. जो ये हमारा शूट अपाइकल मेरिस्टम है, they are having superficial layer and deeper tissue. जो ये हमारी superficial layers है, like L1, this L1 cells are able to form the epidermal layer. जो ये हमारी superficial in shells है, like superficial meristematic cells है, ये क्या बनाती है? ये हमारी L1 cells बनाती है. These L1 cells are actually our epidermal layer. जो फिर इसके अंदर सेट है, जो L1 के अंदर सेट है, they form the L2 and L3 layer. This L2 layer forms the subepidermal layer. जो हमारा L2 है, ये क्या बनाता है? subepidermal layer. और सेल्स है जो L3 बनाती है, वो सेंट्रली लोकेटेड है. अब जो ये L1, L2, L3 है, ये डिफरेंट सेल्स बना रही है. इट हैज बीन ऑब्जर्व दैट द ऑल दिस, मतलब लाइक जिसका क्या फेट है, मतलब L1 का क्या फेट है, L2 का क्या फेट है, L3 का क्या फेट है, इट ऑल डिपेंड्स, और all this is determined by a one mechanism. This mechanism is called as position dependent mechanism. मीन्स कौन सी सेल किस पोजीशन पे है, वो डिटरमाइन करती है उसका फेथ. लाइक हम लोग ऐसा भी बोल सकते हैं, जब हम लोग सेल लीनिएज स्टडी करते हैं, एनालाइज करते हैं, ऐसा देखा गया है जो हमारी इनिशियल सेल्स है, उनका पोजीशन कैसा है in the shoot apical meristem, that position will determine its fate कि ये सेल L1 में है या पोजीशन नंबर L2 पे और L3 पे, तब जाके ये डिफरेंट सेल्स बनाती है. Now, now the factors which are influencing the formation of shoot apical meristem. It has been observed that there is a auxin which is a phytohormone which play important role in the maintenance and the formation of shoot apical meristem. As it has been seen that there is a complex transportation of auxin inside the embryos. Like embryology means seed, inside the seed. It has been observed during embryogenesis there are certain proteins which allow the accumulation of auxin in these apical regions. Like कुछ proteins है जो allow करते हैं auxin को apical meristem में रहने के लिए, and these proteins are called as pin proteins, PIN, pin proteins. And out of these cluster of pin proteins, pin 1 play an important role which leads directly accumulation of auxin in the apical meristem. After there is the embryogenesis, embryo is formed, then again there is a developmental stage. In one of the developmental stage, which is heart shaped, then this pin proteins allow reversal accumulation of auxin from shoot into the root. But these pin proteins always maintain the higher concentration of auxin at the tip. In other sense, we can see that the meristematic cells are having higher concentration of auxin. The role of the auxin has been observed that this auxin does not allow the replication of viruses. So this auxin does not allow replication of viruses in the meristematic cells. That is the reason when we have to go for the virus-free plants for export purpose, that time we always take meristematic cells, as these meristematic cells are having higher concentration of auxins. There are also certain genes which are involved in the maintenance of the SAM, shoot apical meristem, or we can say the constant size of this SAM shoot apical meristem is controlled by genes like WUS genes, which maintains the identity of apical initials or apical meristem. It has been observed if there is a mutation in this WUS gene, then the apical initials, which always remain as a meristematic cells, are then differentiated into the central zone and block ability, or they lose their meristematic activity, as meristematic cells are actively dividing cells. So these, if there is a mutation in this WUS gene, then these apical initials lose their ability to divide continuously as they used to do in case of meristematic cells. Then these cells are getting differentiated into the different zones like the peripheral zones. So this WUS activity is promoted, also maintained by or promote the activity of apical initials by different set of genes. So these set of genes are called as clavata, CLAVATA, clavatas, club shape. There are so these are different set of genes. Clavata is the different set. It includes a different set of genes. So the individual set of genes like clavel 1, CLV1, clavel 2, and clavel 3. The role of the clavel 1, 2, and 3 has been observed first in the plant like Arabidopsis thaliana. It has been scientists have observed if there is any sort of mutation in these genes, then again the then again the apical initial, which always maintain the size, which continuously give this meristematic cells out, but they maintain their size, then this apical initials get enlarged, and it has been observed that these genes act interdependently. It has been observed that these genes act interdependently. If we talk about the arrangement of leaves or the phyllotaxy, like how leaves are arranged on the shoot, depends on the initial pattern of leaf primordia which are present on the shoot apical meristem. As in most of the cases, it has been seen that there are three different commonly pattern observed for the arrangement of leaf on the stem. First, as you can see in the diagram, is alternate, another is opposite, and the third one is the spiral pattern of leaf arrangement. These arrangements depends on many intrinsic factors that are that are able to produce this characteristic pattern of leaves, and it has been seen this characteristic pattern of leaf arrangement is species specific, and there are certain, even they are also affected by the internal patterns, or if there is any sort of mutation inside the meristems, then there will be the alteration of the shoot apical meristem, that again will lead to the different development of leaf arrangements. Again we can say here that it all the like leaf phyllotaxy also depends on the position dependent mechanism. Whatever the cells are there aside, they will decide their fate. This is how the shoot apical meristem is formed.